Okay, so hello everyone. Welcome to today's Immune Zoom seminar. So it's my great pleasure to welcome today's speaker, also my friends, Dr. Penghui Zhou from Guangzhou University Cancer Center. Just a very brief introduction about Penghui. So we actually share quite a bit of the postdoc training experience back in Boston. And then currently Penghui is a professor in Sun Yat-Sen University Cancer Center. Penghui actually finishes undergraduate education in Jiangxi Agricultural University, and then further to pursue a master's degree in biochemistry and molecular biology in Huazhong Agricultural University. And then further, he continued his PhD training in University of Michigan in US, following which he wanted to pursue further study in immunology and cancer immunotherapy. So he moved to Dana-Farber Cancer Institute, Harvard Medical School in Boston from 2009 as a post-training fellow, and then move on to instructor also in Dana-Farber. And then from 2014, he moved back to China and then started his independent research group in Sun Yat-Sen University Cancer Center. So Penghui's group actually particularly dedicated in studying translational research in cancer immunology. And then in particular, he studied interactions between immune cells and tumor cells within the tumor microenvironment. His lab tried to identify tumor-specific immune cells, particular tumor antigen-specific T cells. And through these studies, his lab also tried to illustrate the mechanism or machinery for tumor cell, how they escape from immune surveillance, and how to identify a novel treatment target during the tumor immune cell interaction. And then in order to accomplish that, he his lab tried to establish their personalized TCR-T cell therapy targeting Neoantigen and Tumor Antigen. Some of these work actually currently getting to already phase 1 clinical trial. And then with all these achievements, both during his postdoc PhD training and then also his independent work, he get numerous awards and honors, including the Travel Award from Roche Nature Medicine Symposium on cancer immunology and immune therapies. He moved back in China as Thousand Talents Program, and then he also awarded as Guangdong Leading Talents Program in 2016. I think today Penghui going to talk about his recent work on define tumor antigen-specific T cells during the TCR-T cell therapy. So without further ado, please go ahead, Penghui. We are looking forward to your talk.
All right. First, I would like to thank Tran for providing such a good opportunity to share our recent study on the personalized TCR-T cell therapy. Yeah. So today I'm present. We have done on the basic research to find these tumor-specific T cells and also move them into the clinical trial to treat patients which has no options for current therapies. So first, I would like to go through the cancer immunotherapy circle in the patients. And for any immunotherapy, the tumor antigen is the first step to initiate an anti-tumor immune response. And then we know that the tumor antigens in the cancer cells are mainly from the mutations and also some overexpressed proteins. The APC cells usually is the macrophages, and also the dendritic cells, they will engulf this kind of a mutated or on abnormal tumor cells, and then they will present this antigen in the lymphoid organs, such as draining lymph nodes. And so that T cells recognize these antigens, will be activated, and then they will exit the lymph node and migrate into the tumor site. And over there, they are going to recognize tumor cells and attack them and eventually destroy the tumor. Okay, that's a simple circle for the anti-tumor immune response. And we know the tumor antigen initiate this process. And for the tumor antigens, we know there are two types of tumor antigens. One is tumor-specific antigen, and one is tumor-associated antigens. Yeah. And for the tumor-specific antigens, they are mainly from the mutated genes, such as neoantigens. We now give this name. And some driver mutations, such as P53 and P10, these kind of mutations, we know they are shared in patients. But they have low immunogenicity. Okay, but for the neoantigens, which is randomly mutated, they are not shared among patients. Based on their WES sequence, the whole exome sequence, we know there's a lot of mutations happening in patient tumors, but very rare they will share between patients. So targeting neoantigen actually is quite difficult because they have to be a personalized therapy. Okay. That's why we are currently using a lot of tumor-associated antigens for the targeted to T-cell therapies. For example, the carcinoembryonic antigens, testis antigens, and oncoviral antigens. Most of them actually have no mutation, but they highly express in tumor, also present in the normal organs and the tissues. And then in this way, they will cause not just attacking the tumor cells, they may also attack the normal tissues and cause the side effect. So if we go through the current T-cell therapies, the two major types, CAR T cells and the TCR-T cells. Okay, for CAR T cells, the surface antigens are mainly the targets like GPC3, CD19, and then they're all not mutated. Okay, it's very rare mutated genes on the surface. Okay, and for the TCR-T therapy, they are more antigens in the choice. Okay, because the genes expressed in cancer cells, they all can be presented by the MHC. That's why TCR-T cells can targeting either inside of the tumor cells on the surface or secrete the proteins. They all can target them once they're antigen. That's why the TCR-T cells have more choice for the antigens. But so far, all of them are using majority of them are using the tumor-associated antigens. Because if you want to design a CAR T therapy or TCR-T therapy, you want some patients express them, then you can use them. So if we check all the TCR-T therapies in the past decades, actually you can see most of them are the tumor-associated antigens. And of course, we also see there are some death case reported, like GP100, they have the heart toxicity, and the EGFR also cause the lung toxicities. So targeting the tumor-associated antigen, actually, they have high risk for the side effect. Okay. And in that way, actually, recently, the neoantigen vaccine has been developed and moved into the clinical trials. Okay, so neoantigens, we know they are derived from the tumor mutations, but not all the mutations will become a tumor antigen. Okay, to be a tumor antigen, actually, they need to express into protein, and the protein levels must reach a certain level, so the tumor cells can present them out. And also they must be processed, and then they bind to the MHC molecule. And finally, they must have T cells can recognize this kind of antigen. Okay, so if we define a neoantigen, the basic concept must be there are some T cells can recognize them in the patient, right? But currently, for the neoantigen vaccine, we always use the avidity of this antigen to the MHC molecule to predict them. But this is only one part of that. That's why the prediction accuracy is quite low. Currently, only 20 to 30% of the neoantigens are accurate of the prediction. And we can check recent papers published in like Nature and Cell, several journals for the clinical trials data as well. And if they immunize the about 10 peptides, only like 2 or 3 peptides will generate enough immune response. Okay, and in another way, because immunization will not generate enough T cells. Okay, that's why when with the T cells went into the tumor environment, they will also be suppressed and cannot generate enough in the tumor response. Okay, so we can see that the neoantigen vaccine do show some therapeutic effect, but overall their efficacy is quite low. Okay, so in our point, we are trying to see if we can identify the TCRs which recognize these kind of neoantigens, right? Because neoantigen is harder to predict, but we know every neoantigen will have T cell to recognize them. If we go through the T cells and find the TCR, then we may be able to get a TCR, clone the TCR, and generate the TCR-T cells. Okay, and these kind of TCR-T cells will specifically targeting the neoantigen. Yeah. And we think that might be a better way to attack the tumor, and it might have a better efficacy on that. But currently, we don't know which kind of cells are tumor-specific T cells and which one is bystander T cells. And if we look at the tumor, we know that the macrophages and the DC cells, they carry the tumor antigen and activate the tumor-specific T cells in the adrenal lymph node or other lymphoid tissues. Okay, and this kind of T cell will migrate into the tumor. That's what we learned from the tumor immune response. Okay, but at the meantime, we know that the blood vessels will also infuse a lot of bystander T cells because there's a lot of blood circulation in the blood, right? And so in the blood vessels, there is a lot of like a naive memory and all kinds of T cells. They will also migrate into the tumor. In that way, the tumor is composed of bystander T cells and the tumor-specific T cells. So first, we need to identify which one are the tumor-specific T cells. Then we can gather the TCR to use them for the TCR T cell therapy. Okay? So the first thing we needed to finish is to identify the tumor-specific T cells. So as we know, the tumor-specific T cells, they must experience the tumor antigen stimulation locally in the tumor. So the expression and also the TCR expansion might be different from the bystander T cells. In that way, we designed these experiments. First, we got all the samples from tumor, peri-tumor, normal, and blood. And then we got the T cells, perform the single cell sequencing and also the TCR sequencing. In that way, we can cluster the different cell populations, and we also can characterize the TCRs in these subpopulations. And in the meantime, we also can clone the TCRs to generate the TCR-T cells using their own PBMCs. In another way, we need to get the tumor antigens because the only point to prove they are tumor-specific T cells, they must recognize the tumor antigen. So in that way, we also did the whole exome sequencing and the RNA sequence of the tumor. And we find all these kind of mutations. There are a lot of mutations, as we know, is the patient that we sequenced, they have 300 to 500 mutations. We don't know which one are tumor antigens, right? And because the prediction is quite inaccurate, so we pick up the top 100 mutations and composed them into tandem genes. Because every mutation they can recognize if they can recognize by the MHCs, there must be like 10 to 14 peptides on the MHC class 1, right? So in that way, we pick up the mutated amino acid sequence and we put the mutated amino sequence in the middle. And on the left and the right, we add another 12 amino acid in that. And totally there will be 25 amino acid peptides. And then we synthesize these kind of peptide genes and link them together. So in one vector, we can actually construct around about 30 amino acids, this kind of peptide in there. And of course, we need another antigen presenting cells present them. So B cells are very good antigen presenting cells. They have high expression on MHC molecules, but they cannot culture well in vitro. In that way, we infected them with the EBV, and in that way, we can convert them into lymphocyte blast. In that way, we can culture them in vitro for a long time, right? And then we will transduce these kind of antigen genes into the LCLs, and then we will get these kind of antigen presenting cells. They express all the tumor antigens, right? And because we also generated the TCR-T cells, then we put them together to see which kind of TCRs will recognize this antigen. Yeah. And after stimulation, then we got the TCRs recognize antigen and not recognized antigen. Then we will go back to check where are those TCRs located in these cell clusters, right? And in that way, we actually can trace back which clusters are tumor-specific T cells, right? That's how we characterize the tumor antigen-specific T cells. And in another way, we also generated the PDX models from the same patients. And we also use these kind of TCR-T cells to treat these patients, the PDX models, to see if the TCR-T cells developed by this approach can actually treat them, right? That's the whole design of the experiments. And let's check the data we have. So first, we divided the CD4 and the CD8 T cells into different clusters. And as we can see here, most of the traditional populations, such as naive T cells, memory T cells, effect T cell, exhaust T cells, and Tregs, they all can be identified. And also some special, some different clusters, like some cells with high metastasis cells were also identified, right? And based on these clusters, we further checked if they, which one are enriched in the tumor, okay? Because we already sequenced the tumor, peritumor, normal, and blood, then we can trace them, okay? And the reason we sequence all these kind of samples is because we wanted to see which one are circulating among those tissues and which are actually specifically expanding the tumor. So as we can see here in the CD4 T cell population, we found the two clusters, they are enriched in the tumor. And in the CD8 T cells, we also identified two classes over there, right? And there are some clusters actually we can see they shared among peritumor and the normals. So they might be this kind of circulating bystander T cells, but we have no direct evidence. So we further characterize these clusters. Okay. Then we check the TCRs. As we know, if they are expanded by the tumor antigens, then the TCR distribution among these tissues might be different. Okay. So as we can see in the figure A, we can see there is there are some TCRs actually share among tumor, peritumor, normal, and blood. So this indicates this kind of TCRs are circulating among these tissues, right? And also some of them share among 2 or 3 tissues. But in the meantime, we also find some TCRs are specifically present in the tumor only, right? Both happening in the CD4 and the CD8, right? That means that some TCRs do happen, and they were expanded by this kind of tumor antigens, right? So we will check which kind of clusters contain this kind of TCR with specific expansion, right? Then we check the TCR distribution among all these tissues in these subclusters. As we can see in the right side, there are 2 clusters in CD4 and 2 clusters in CD8. These 4 clusters, as we previously identified, they were enriched in tumor. But when we check the TCR frequencies, you also can see that the frequencies are highly expanding in the tumor. but not in the peritumor normal or blood. And some of them do have a little bit of expansion in the peritumor because peritumor is around the tumor. So that's quite reasonable. But for other clusters, you can see this kind of TCR, actually they distributed in all these 4 tissues. That indicate that they are actually circulating around among these tissues. Okay, so because the tumor antigens activated this kind of tumor-specific T cells. Okay, so we also analyze the TMB and the percent of these clusters to see if they are correlated in the tumor tissues. Right. As we can see here, again, this kind of 4 tumor-enriched clusters, they're positively correlated with TMB, right? So here are 10 patients. We did the single-cell sequencing and also performed the TMB sequencing. And we do find that they are correlated with the tumor antigens. So that means they might be expanded by these mutated neoantigens. And next, we want to find which kind of genes are specifically expressed in these tumor-specific T cells. So we pick up these tumor-specific T cells and the shared T cells, compare their gene expression in CD4 and CD8 T cells, and also CD4 Tregs. Okay, as you can see here on the figure B, you can see like some genes are specifically in the CD4 T cells, CD4 specific T cells, right? And some of them are CD8 tumor specific T cells. But we found the CXL13 is a very unique gene, actually highly expressed both in CD4 and CD8 tumor specific T cells, but not in Treg, okay? And also other shared populations. So in this way, we actually identified a very specific marker for these kind of tumor specific T cells. Yeah. And we also find that some CD4 Tregs, they actually have a lot of markers shared with the tumor-specific CD4 and the CD8 T cells, indicate they all have experienced tumor antigen, right? And the next, we would like to check if this kind of CXL13 cells actually have the characteristics of tumor-specific T cells, right? So we also checked the expression of this of CXL13 in the clusters we identified by the single cell sequencing, right? So you can see here the tumor enriched clusters there are 2 clusters in CD4 and 2 clusters in the CD8. They all highly express CXL13, right? And once we check the TCR expansion, we also found both the CD4 and the CD8 CXL13 positive cells. Their TCRs are specifically expanded in the tumor, but not in the peritumor, normal, and blood. In contrast, the CXL13 negative T cells, their TCRs are actually shared among those tissues. So that TCRs must be circulating among those tissues, right? So in this way, also confirm that the CXL13 is a specific marker for these tumor-specific T cells. Okay. And we also checked the correlation with the tumor mutation burden. Indeed, the CXL13 positive cells, they are positively correlated with the TMB in the 10 patients we sequenced. We also checked the TCGA data, and we checked we calculated the tumor mutation burden and also the median expression of CXL13 in all kinds of TCGA tumor types, and then did a correlation. And it also shows that the CXL13 positive expression, CXL13 expression are also correlated with tumor mutation burdens. So in this way, both our data and the TCGA data consistently show that the tumor mutation burden are correlated with CXL13, right? This means that the CXL13 cells are very likely be expanded by this by high tumor mutation burden tumors. Right. Of course, we need to prove if these cells are recognizing tumor antigens. So we go back to the tumor antigen stimulation system in vitro, right? So as we showed before, we synthesize these tandem genes or tumor antigens, okay, and then transduce them into the B blast cells and then turn them into the tumor antigen cells. And we also pick up the top TCRs from CXL13 positive cells and the CXL13 negative cells, and then culture them with the antigen presenting cells to see if they can be activated. So as we know, the activated T cells will express interferon gamma. So we're using the interferon gamma expression levels as the standard to identify if they are recognized these kind of tumor antigens, right? So as we can see here in the CD4 positive CXL13 positive cells, we tested 5 TCRs. We found 1 TCR recognized tumor antigens, but the negative ones, none of them recognized, right? And in the CD8 CXL13 positive cells, we also tested 5 of them, and 4 of them are positive. And the negative ones, none of them recognize tumor antigens. Right? So in this way, we directly proved that the TCRs from the CXL13 positive cells, they actually recognize tumor antigens. It also relies on the TCR frequency. Okay, so in those patients, we found the CD8 positive CXL13 positive cells, they have high frequency of TCRs, but for the CD4 positive cells, they actually have low frequencies, and that might be because CD4 T cells was not well expanded in the tumor, and we also didn't include all the tumor antigens. Because like this sample, these patients, we sequenced we found that there are more than 500 mutations, but we only included the top 100. So it might be some tumor antigen was not included. So that's why we cannot find all of them are recognized the tumor antigens. Okay, whatever we find that the CXL13 cells is actually recognized the tumor antigens. So the most important thing is if they can treat the cancer, patient derived xenograft. Okay, so as you can see, the CD4 positive, CXL13 positive cells, the TCR we found, they recognize the tumor antigen. Yes, it actually can treat the cancer, can treat the tumor. As you can see, the tumor was largely delayed for the growth in the PDX model. And for the CD8 T cells, they actually showed much better efficacy on the therapy. You can see all the 3 TCRs we picked up, they all showed quite a significant therapeutic effect on these PDX models. So that data proved that if we can obtain these TCRs from the CD8 T cells or CD4 CXL13 cells, they might be able to use to treat these patients. Of course, the frequencies is also another point. We need to decide which kind of TCRs you are going to use. Okay, because every time if you sequence a patient, you will find more than like 10 TCRs over there, and most of them have different frequencies. And in that way, you need to decide which one you're going to use. So in our future work, we'll also try to find which one is better. It all depends on the frequency or the gene expression levels. In the future, we are going to figure that out. Okay. And the next questions we asked is, if this kind of a CXL13 tumor antigen specific T cells exist in all these kind of tumors, right? Because we only detected in the non-cancer patients. So we also analyzed all the available published single cell sequence data. And we also analyzed the subclusters in this kind of published data. We found all of the tumors, we can identify the CXL13 population. And if we check the other genes of the CXL13 populations, they are actually consistent with what we found in the lung cancer patients, right? And this means that the CXL13 positive tumor TAS T cells, they present in all kinds of tumor, right? And the T cells might experience similar processing once they are experiencing the antigen stimulation in such a tumor microenvironment. Okay. And because the CXL13 is an intracellular marker, right? It's an intracellular protein, then it's hard for us to sort them by FACS. Okay? Then we're trying to establish an approach using FACS to sort them using the surface markers. As we showed in the showed before, the gene expression data, except for the CXL13, we'll also find some other genes, like in the CD40 T cells, CD200 is a marker, although not that specific, but they're also associated with CXL13 positive T cells, right? And in the CD8 T cells, we found ENTPD1 is also a specific marker for them, right? So we then use these kind of specific markers on the surface to sort them. And once we sorted them out from the tumor, we also did a qPCR to see if they express the CXL13. Yeah, and indeed we do see they have highly expression of the CXL13 in these sorted T cells. And we also activated them using the tumor T cells. Okay, not tumor, using the tumor cells, right? So once we activate them in vitro, we also can see this kind of sorted T cells that have higher interferon gamma expression. So in this way, we actually can enrich this kind of T cells from the tumor by using these surface markers, right? And of course, we will have to using the single cell sequence to detect the CXL13 expression and the TCR sequencing as well, right? So based on what we have found, we establish therapy for the personalized TCR T cell therapy. Okay? And we also moved that to the clinic trial in our hospital. So here is the processing. First, we will gather the tumor from the patients, either from biopsy or their surgery. And once we gather the samples, we'll process them and sort them by fax. And then after that, we will using single cell RNA sequencing and the TCR sequencing to find the specific TCRs which can be used for the patients. Yeah, and we then clone these kind of TCRs and produce virus, transduce them into the T cells isolated from the PBMC from the same patient, and we will then expand them for about 2 weeks, and finally we'll reach about 1 billion T cells, and then we can infuse them into the patients for the therapy. So as we can show here, in about 6 weeks, we can finish the whole processing. Of course, we already modified the whole processing to make it smoothly and in time to produce enough cells for the patient. Okay? And right now, the clinical trial is recruiting patients as well. And Dr. Xiaoshi Zhang, who is the PI of this clinical trial, is in our hospital. Okay. Here I show one example of the patient. And this patient is a melanoma patient with non-metastasis. So we got the sample from the surface tumor, and then we purified this kind of T cells and generated personal TCR T cells. And as you can see, the metastatic tumors in the tumor has been shrink quickly once we infuse the T cells back to the patient, right? And prove that this kind of therapy actually is a potential approach for this patient who has no choice for other therapies or failed all the therapies. Okay? And we also have observed a very secure secure data from these patients, right? And we didn't see this kind of grade 3 side effects. Most of them only showed grade 1 side effect on that. Right? In another way, we also use these T cells to predict the response to immune checkpoint blockade. The reason we do that is because we think that the CXL13 cells, they recognize tumor. Okay, and if we using ICB therapy, they must be to block them and then make them recover them from the patients and then use them to treat the patients. Right. So it is very important for these levels. And then we can see here is if we using this CXL13 for the for the IHC, we can actually very accurately identify them on the tissues. Right. In that way, we actually can measure their levels quickly and very accurately. So we're using this IHC approach to determine the TAS T cell levels in all the tumors. and also trace back for their therapeutic efficacy. And what we can see is here is both in melanoma patients and in the CRC patients, the CXL13 positive patients showed a pretty good response to the immune checkpoint blockade. And actually, it's much better than the prediction accuracy of the PD-L1, okay, which is widely used in current clinical for the prediction. Okay? And also, we are testing other tumors as well. I didn't show the data here. We are currently recruited more than 1,000 patient samples, and we will test if this CXL13 is a very wide marker for predicting the immune checkpoint blockade. And of course, in the future, we are going to include some other markers like PD-L1, also include that to see if we can get the best composition of these kind of markers to have the best prediction effect on the ICB therapy. Okay. So I would like to summary for this personalized T cells compared to the traditional therapies. Okay. So as we can see here, if we use this personalized T cell therapy, it's not limited to currently known antigens. For example, if we use NY-ESO1 TCR-T cells, first the patient needs to express NY-ESO1 in the tumor. And in the meantime, the MHC subtypes also need to match this kind of TCR. Okay, we know every TCR match to a specific MHC molecule, right? So as we know, the NY-ESO1 TCR currently used in clinical recognize the HLA-A0201. Okay, and this HLA subtype expressed quite low in Chinese patients. Okay, I remember it's about 50%, 5% to 10%. Okay, and if you also require they express NY-ESO1, probably only I was less than 1% of patients will be qualified for this kind of therapy, right? So as we know, this kind of limited expression and MHC restriction largely inhibited the clinical use of this TCR-T therapy, right? But if we use this personalized T cell therapy, we don't need to know any antigen, known antigen over there, because the patients will respond to its own neoantigens. And even don't need to know that which neoantigens they recognize. You only need to purify this kind of T cells from patient, then you can use them. Okay, so the personalized TCR-T cells actually has a very wide indication. They can be used in majority of solid tumors. Yeah. And the second, because we can use multi TCRs to target more antigens, so they must have much higher efficacy. Right? So traditional TCR-T therapies, we only have one TCR to recognize one antigen, and we cannot guarantee all the tumor cells express the tumor antigen. So it's quite difficult to target all the tumor cells, and of course, it will cause the escape of these tumor cells don't express the antigens, right? But if we can use multiple TCRs to target multiple antigens, we probably can cover most of tumor cells and then avoid this kind of escaping, right? And the third part, because we are targeting tumor antigen, which is specific in the tumor, right? We call it the neoantigens. So they were not targeting the normal tissues. So of course they have a better safety. That's what we have observed in the clinical trials. We don't see much side effect for this kind of infusion, right? And of course, the last one is the timing, right? For any specific or personalized TCR-T therapies, you have to supply to patients in time, right? As we know, there are several approaches being used in clinical or in clinical trials, but we don't see any of them can finish in 2 months. And for the patients at the advanced stage, if they need to wait like half a year, of course, it's not good for them. They probably already died when the T cells are ready, right? So we actually optimized all the processes in this kind of TCR-T cell therapy, and finally we can generate them in 2 months, right? And we're trying to improve them in the future. And our goal is to generate these kind of T cells in 1 month, right? And in that way, we actually can apply this kind of therapy for patients who are at the very advanced stage, right? They have no choice for other therapies. Yeah. And for the last conclusions from this, from our studies, we actually identified T cells in majority of cancer patients, right? And we also using, we also found the CXL13 is a specific marker for TS cells. And with this specific marker actually can help them, can help us very quickly to obtain these TCRs, recognized neoantigen, right? So in that way, we actually can use this marker to gather the TCRs and quickly generate this kind of TCR T cells. You know, in the PDX models, we show they have very good efficacy on these models. And now in the patients, we also observe some good response. Of course, we are expanding our patient indications, okay? Because currently we only included 2 or 3 indications, and in the future we are trying to include more of indications like other tumor types. Okay, and overall, we're using this approach. We can target in the neoantigens, which has never been targeted by TCRs in the past, right? And also, this kind of a personal TCR T therapy, they actually have a lot of advantages than the neoantigen vaccines. Okay, and so overall, from this research, we start from the basic immunology idea that the patients must respond or they have T cells recognize their antigen. And then we then use this kind of T cells, generate the personalized T cell therapy, and move them into clinical treated patients. Hopefully, we can expand this kind of clinical trial in the future and use them in more patients and in more indications. Okay, and this work actually is quite hard because we need to collect a lot of samples and also need to define every single TCRs. So we have a lot of students and postdocs is involved in this project, and also including our clinical doctors, Dr. Hao Jingjing, Dr. Xiong Xingxing, and Dr. Liu Zhenjiang. They are my postdocs in the lab, and Dr. Yang Han, Li Dangdang, and Zhang Xiaoshi. They are clinical doctors from our hospital, and Dr. Li Wende is from our animal facility, and Shuangyan is my students, one of my PhD students. They did a wonderful job on this project and make it happen. Yeah. And finally, I would like to thank you all for the attention. Yeah.